I think so.
Okay, I'm gonna start just a minute early today, give you a quick update. So last week I talked about my daughter who had this kind of suspicious bug bite. She went to the doctor and they put her on doxycycline because they were afraid it might be Lyme disease. So she was on a tetracycline, and we talked about how tetracycline has these kind of strange side effects last time. So that's why we don't give tetracycline to kids. It affects growing bones, growing teeth. That really ugly picture I showed you of some teeth that were discolored by tetracycline. It's phototoxic, and a lot of antibiotics carry the side effects of giving patients diarrhea or vomiting. So I was a little worried about my daughter. She was running the riverbank run this weekend, a 15-mile race. Thumbs up. It went great. So woohoo! She flew in from, she's a part of the Georgetown Running Club. So that's not GRCC, it's from Washington, DC. So thumbs up. She did throw up at the end of the race, but probably not because of the doxycycline. So she was pretty happy. So that was fun for us. I hope you all had a chance to relax for a little bit over fall break. Some of you are shaking your heads. No, it just makes it harder to start up again. I know. I am having some additional office hours. I'll be in my office tomorrow morning. I'll probably be there before 10, but at least from 10 to 12. So if any of you want to come in, talk about anything, our next exam is a week from today. So that's going to cover chapter 9, antimicrobial stuff, 10, antibiotics. And we are going to start today, chapter 14, process of infection. And hopefully we're going to get most of the way through that. I think I'm going to finish that up on Tuesday, but it's just some kind of definitions mostly that we'll finish up with. So I'm going to try my best to kind of finish up our antibiotics, get moving, and then still have that exam next Thursday. So we finished up last time talking about antibiotic resistance. And this can be rather complicated to kind of think through. We had three kind of a fourth method that organisms acquire the ability to withstand antibiotics, so to be resistant or grow even though antibiotics are present. Okay, so the first mechanism we talked about was that they just limit the access. Okay, they don't allow the antibiotics into the cell, or if it does come into the cell, they pump it right back out again. All right, so alter the porin structure. Remember, the porins are those channels in the outer membrane of gram negatives. Alter that so that the antibiotic can't get in to begin with, or once it does come in, they have a way to pump it right back out. So that was the first one. And the second one we talked about is that the organism acquires the ability to alter the antibiotic. Okay, so that's if they acquire the ability to make an enzyme like beta lactamase, then they can, cleave part of that structure of the drug, and then the drug is no longer effective. Okay, so beta lactamases are one example of that. There are other examples that we have found of bacteria able to acquire enzymes to, again, alter some structure. Maybe it's just some kind of functional group. Maybe it's just adding a phosphorylation or phosphorylating an antibiotic in some way. So it doesn't bind where it needs to be able to bind in order to work. Third mechanism. Okay, and I don't think we got to this last time, so this is kind of where we're picking up from last time. Oh, it looks like my slide got scrunched together a little bit. Sorry, I must have forgot to save that in the right way. Some bacteria acquire the ability to actually modify part of their own structure so that then the antibiotic can't bind in the same way. So it's a little bit harder to wrap your mind around. You would think that that would alter their viability maybe by doing that, but sometimes not. So we found a couple of examples of that. Some bacteria can methylate their ribosomal RNA. So there are a lot of antibiotics that bind to, say, the small subunit maybe, or the large subunit of the ribosome. And if the bacteria modify themselves just a little bit, and maybe it's just by adding a methyl group, then the drug can no longer bind. Okay, so keeps the drug from binding. Okay, it can no longer interact properly with the ribosome, and so the antibiotic becomes ineffective. We found cases also where organisms have been able to do just a very small mutation, maybe even just a 1 base pair mutation in one of their enzymes. So for example, if they can alter their DNA gyrase just slightly so that a quinolone drug or fluoroquinolone can't bind the same way, then those antibiotics are going to be ineffective. The gyrase still works. Okay, 1 maybe base pair doesn't, maybe in a spot that it doesn't make any difference. Or same thing, RNA polymerase is big enzyme. Maybe you just change 1 little thing, maybe 1 little amino acid different, and that may alter the ability of rifampin to bind to that. Okay, so sometimes very small changes can have big repercussions. All right. The fourth mechanism is not really a mechanism quite per se, but we know that some bacteria acquire the ability to be tolerant. And they often do this when you have like kind of big communities of bacteria growing together in a biofilm. And sometimes when there's big mats of them growing together, they're able to switch their metabolism off a little bit, and they just kind of persist. Okay, we call them persister cells. So they're not actively growing, they're not metabolizing very much, they're just kind of, surviving. Okay, and bacteria are really hard to kill when they're like that. Most antibiotics only work, or a lot of antibiotics only work when the cells are actively growing. So it's very difficult. This might happen maybe when a patient has an infection on some kind of implant, and you get a biofilm formation on an implant. Really tough to get antibiotics delivered to that spot internally where they're going to do any good. And they end up having to just operate and take out the implant when you have an infection like that can't be controlled. I did just want to point out that there was a very interesting article that I read. It's been a few years ago now, 2017. There was some research done at MIT where they were trying to jumpstart cells that are in this persister mode. So how do you get cells to start actively growing again? Same way, say you have a kid that's sick and won't eat anything. Well, you don't offer them broccoli, right? You offer them a popsicle, and they're more likely to eat something when it's something attractive to them. So the same way, the researchers have provided bacteria in these persister states with not only a sugar source. They found that wasn't enough, but they also provided some kind of electron acceptor for them to kind of jumpstart their metabolism so that they would be more susceptible to antibiotics. So just kind of an interesting, really brief article there. All right, so that's pretty much, antibiotic resistance. This was really the #1 health emergency before COVID came along, and it's kind of got stuffed back down again. But this is one of the things that, infectious disease specialists keeps them up at night worrying about, are we going to have organisms that we don't have anything to treat anymore? Okay, so they very often acquire the ability to become resistant either by mutation or by some kind of horizontal gene transfer, most often conjugation. All right, so this is a little exercise that sometimes I do in class. I don't want to take the time to do this right now. You can look at it later. These are 5 kind of schematic describing those different mechanisms. So take a look at this later on. I'll do one with you here, number 3. This is describing an efflux pump. So the antibiotic comes into the cell and the bacteria has a way to just get rid of it right away again. Okay, so look at that later. I can post which number is which if you want to check your answers, but it's a nice little exercise to just kind of get those 3 mechanisms kind of straight in your mind. All right, any questions, comments about antibiotics before we move on? All right, good. Process of infection, chapter 14, hopefully some information that you will find applicable to your life right now, living in a pandemic. And it's a good introduction to, as next week we'll start talking about immunology and some of the ways that we respond to infection. Okay, but chapter 14 is just describing infection, describing some of the tools that bacteria have to be successful, some of the terminology we use as to how we pass microbes around between each other. Okay, so we're going to start with a little discussion of the normal microbiota already in our body. We'll talk a little bit more about that in terms of our immunology chapters, but we'll just start there. Thinking about, we'll talk some terminology as well. Portals of entry. How do microbes gain access to the body? What's the most common ways they do that? And then kind of following the process of infection, usually those microbes have to attach to a certain kind of cell that they're specific to in the body. And what are some of the mechanisms they have to be successful doing that? Not only attaching, but invading maybe into our cells as well. How do we describe that whole process in our body? I'm just going to touch on that real briefly. And then microbes have to be able to leave a host to infect another host. So portals of exit there, and talking a little bit some epidemiological things. How are they transmitted to other hosts? And again, it's kind of a lot of terminology that we use that you want to get familiar with. All right, so first, just have to mention our normal microbiota, because when we're thinking about being infected by, disease-causing organisms, we have to reflect that we have a lot of cells in our body that are not our own eukaryotic cells. We have at least as many bacteria, not only bacteria, but things like fungi, archaea, protozoa, and even viruses, we think, that live in our body just normally. And they're not causing any harm to us. They probably help us in a lot of ways. Okay, so that's what we call our indigenous microorganisms, or our normal microbiota. We have at least as many of those microbes in our body as our own cells, probably maybe even like 10 times more microbes than our own cells. So they are part of who we are, you could say. So the normal microbiota, those are not the organisms that are typically hurting us or causing us disease. Usually we describe their relationship to us some kind of symbiotic or at least synergetic relationship. So if we describe our relationship with them as mutualistic, that means that both the microbe and us are both benefit. Okay, so both organisms, meaning us as the host and the microbe, benefit. Okay, so we give them a nice warm place to live with lots of nutrients. And pretty often, we're learning more and more about this, they will provide some kind of benefit to us. Maybe it's helping us to digest our food, or maybe it's just outcompeting other organisms that would be more likely to cause disease. Sometimes we can describe the relationship as commensalistic, meaning that one member benefits and the other member is not affected. And sometimes it's a little bit hard to tease out which of those two relationships it is. For a long time, we thought the staph epidermidis that lived on our skin was a commensal. Okay, we provided nutrients for it, and it wasn't doing anything for us, but it didn't harm us. Well, we now know that we've understand ways that it does actually help us. So sometimes the more we learn, then we adjust our thinking. Okay, so we kind of classify our normal microbiota. Sometimes I slip and call it normal flora. They decided a few years ago, don't call microbes flora because that makes them sound like they're plants. Okay, so I have to say microbiota instead. I always kind of trip over that. We can describe that microbiota in two ways, either as being transiently associated with our body, and that just means that it's here for a time, and then we lose that population of cells for whatever reason, or they're residents, meaning we have acquired those microbes at birth, or maybe during birth, or maybe soon after birth. And we basically have that population of microbes our whole life. We know there's a good bit of difference between individuals. We've tried to quantify and qualify one person's microbiota with another, and we know there's some similarities, but we often find that there are differences as well. We have a long way to go in understanding what are the right microbes that each person should have in order to make them healthy. It's a really exciting area of research right now. Your book lists some examples of resident microbiota, not to memorize at all, but there is definitely differences in the kind of bacteria, for example, that we have in one part of our body versus another part. Like the bacteria that are in our large intestine, for example, it's mostly an anaerobic place. So we have mostly anaerobes, and some facultative organisms. Or that's going to be much different than what we find on our skin. So just to point out, even like in our upper respiratory tract, you might have some candida, some fungi, lots of other bacteria. Here, protozoa is a common member apparently of the upper digestive tract. But there are sites considered azenic in our body, meaning no microbes. At least for now, we consider some of the internal organs, the central nervous system, the blood, the lymph, those places shouldn't have any microbes in them. And if there are, then that's probably causing us a problem. It's probably causing us some disease. Okay. And maybe in the future we'll find out, we'll have better ways to detect them, and we'll find out that that's not the case, but at least for now. All right, so I want to talk about a few terms, and we'll try to use those correctly. One thing, the difference between the terms infection and disease. This kind of seem like about the same thing. Technically, an infection is just when we have some microbes colonizing our body, okay? They may be causing harm, they may not be causing harm. You could argue that we have normal microbiota. Those are organisms that are infecting us. We don't usually think about it that way, though. As to why our immune system allows those microbes, that's a whole nother story that we don't really know, but we'll talk about that another day. And then when we say, we use the term disease, that typically is describing injury to the host. So it may be by infection, it may be, you know, something else going wrong in our body, but infection doesn't always cause disease. We know that we can have somebody be infected, even with COVID, for example, and they don't even know it. They don't have any signs or symptoms. But very often, disease follows after infection. Okay, a true pathogen, that's either a virus or a bacteria that's causing disease in a healthy host. Typically, if a protozoa or fungi causes disease, we call them a parasite. And that's just a little, you know, we'll try to use those words right in the correct way. But again, a pathogen, no matter how healthy you are, if you are exposed to, say, rabies or, you know, botulism, okay, you can get sick with that, no matter how healthy you are. So a true pathogen can cause disease in a healthy individual with normal immune responses. And then an opportunistic pathogen, that's a virus or bacteria that's only going to cause disease when the circumstances are just right. And we'll talk about what some of those circumstances might be. But if the host is normally healthy, for instance, you're not likely to get a Pseudomonas infection. Okay, that's usually an organism that only causes diseases, again, when the circumstances are just right. Oh man, I forgot to forgot to save my slides. It's chopped a little bit off here, but two terms here, and this should be on your PowerPoints, okay? The difference between pathogenicity and virulence. Okay, these are two terms that I went ahead and wrote out for you because the way the book uses them is different than the way I read about them in every other source. So I don't want to get hung up on the definition, but I want to try to use the words correctly. When we try to measure pathogenicity, that's just, if we use that correctly, that just means whether microbes are capable of causing an infection. All right, so is it pathogenic? Yes or no. This is typically, though I read the correct way to use that term. And then when we measure the virulence, I think I defined this as the measure of disease-causing ability, or something close to that. Okay, and then if we want to know, like, how pathogenic is it, that's really not correct use of that term. The better way to ask is how virulent is something. So when something is if it's highly virulent, then that is a pathogen that's most likely going to cause a disease. Trying to give examples. When I give you examples, I'm not worried that you know anything about that example. I'm just trying to relate it to something that you may have heard of. We will talk about specific infections a little later, in 3 or 4 weeks or so. For now, I'm just giving them as examples. If we want to talk about, there's some organisms that are highly virulent. They have a lot of tools. They're very successful at causing infection. And other organisms are not as virulent. They're not likely to cause an infection. Your book makes the point of saying, just because something is highly virulent doesn't mean it causes severe disease. But I would argue not the case, that if something's highly virulent, that's kind of inferred that it's more likely going to be a severe disease, because we measure virulence based on case fatality ratios. So something that, you know, kills a lot of people that it infects, we consider highly virulent. And if something is avirulent, then it's something that's not harmful at all. So usually when we talk about virulence, something's really virulent, that's a really serious infection. And a virulence factor, that's the other term that should be there on your PowerPoint. That's just kind of the tools an organism has at its disposal to be successful at causing an infection. So we're going to look at some examples of those today, things like toxin production or enzymes that it secretes in order to be successful at establishing an infection. Okay. Sometimes an organism that's part of our normal microbiome can actually end up causing an infection. So I'm talking about an organism that's present in our body. In some cases, when the opportunity or circumstances are just right, it can actually cause disease. So what are some of those opportunities that could cause that to happen? Well, if, say, that skin organism, you get a big gash in your arm, well, now suddenly those microbes that were on your skin can have access to your bloodstream. All right, and then they have the potential to cause disease. A few weeks ago, a friend of mine, her appendix burst. Okay, so that's a medical emergency because now all that normal microbiota that was housed in your appendix now has been released out into your body cavity, and those organisms can, cause tremendous infection, and that's not a good thing. So anytime that those normal microbiota are introduced somewhere else, that can be a bad thing. Anytime the host has some immune suppression, so we can think of a lot of ways that this might happen, that our immune system could get suppressed a little bit. As the host ages, we know that as we get elderly, our immune system suppresses a little bit. Just doesn't work as well when we get older. Okay, so as we age, maybe some kind of genetic defect in immunity, such as, there's all sorts of things that can go wrong, like somebody's not able to make a certain kind of antibody, for instance, then that can make an organism able to cause infection that normally wouldn't. Immunosuppressive drugs. So for example, somebody goes on a steroid to treat mono, or you're on prednisone, which is a steroid. That can suppress our normal immune response and make us more likely to have some kind of infection. Here's a good one: physical or mental stress. Okay, we know that can make us more likely to have infection. If you have some kind of other infection first, like you have a cold, for instance, sometimes that can kind of suppress your immune system, make you more likely to get maybe bronchitis or something from that. You're not eating properly. Okay, all those things can kind of tip the balance, and an organism that wouldn't normally harm you, maybe the circumstance arises. The third thing here, changes in the normal microbiota. We talked about this last time. Say you're on a course of antibiotics, maybe some broad spectrum antibiotic that kind of takes out some of your variety of normal microbiota, and then it allows some organism that was normally there in small numbers to overgrow. Some examples there, again, that chopped off my slide here, but vaginal yeast infections or thrush caused by candida that is normally in your mouth, but in small numbers, it overgrows, or C. diff. That's much more likely in hospitals and nursing homes, but I have talked to students your age that have gotten C. diff just from being on a course of antibiotics. All right, so just kind of overgrows. So, all right, so sometimes even, opportunists can cause problems. All right, other things to think about, and I like this picture here. This, some of you in lab may have had the chance to go around and swab places in the lab or in the hall and streak it on a petri dish, some nutrient agar or something, and just see what grows. I used to let students take dishes home and, swab their counters at home and things. And some students would come back and just be so grossed out. Like, look at all these microbes that are growing, in my sock or whatever. And I tell them, okay, keep in mind 3 things. Number 1, what species of organisms are growing there? These are organisms that you've probably been around your whole life. Stuff on your countertop or whatever. You've been around them your whole life. Some organisms definitely are more dangerous than others, but these are probably not ones that are. Keep in mind the numbers. Okay, you're growing up bacteria in much larger numbers on that nutrient agar than what they are on your countertop. If you got colonies growing on your countertop, then that's a problem. Yes, you should clean it. And infectious dose does matter. So we'll look at that. And if you're normally healthy, then that's something else in your favor as well. So, exposure to a wide variety of microbes is good for our immune system. That's kind of my mantra. All right, so I want to look at each 3 of these factors just kind of briefly. There's a table in your book here that just talks about virulence of some microbial pathogens. So species of organism does matter. It's got the more virulent organisms at the top. So remember, these are the organisms that, again, are more likely to cause an infection. And when they do, it's more likely to be a severe infection. So the example they give is one that's not common, tularemia, also called rabbit fever. Not a very common infection, but if you're exposed to it, it's transmitted by the fleas that live on rabbits, then it's a pretty serious infection. And if you're exposed to it, you're probably going to get sick. Bubonic plague caused by Yersinia pestis. So really successful pathogen, we'll say, historically. And then, whooping cough, a little bit less so. Okay, so these are not to memorize, they're just giving you examples of that Bordetella pertussis. People actually can be carriers of that and not get sick themselves. And if they say are around a baby, then it can get the baby sick. So you can be a carrier without getting sick yourself. Pseudomonas, this is another kind of more of a hospital acquired infection, more of an opportunist. So we're getting organisms that are less likely to make you sick. C. diff, again, an opportunist. Okay, you probably have some as normal flora right now, and it's not causing you any problem. Candida can be considered an opportunist as well. And then at the bottom of the list here, these are organisms like lactobacilli. They're found in yogurt. Okay, so we ingest those all the time, and they don't ever make us sick. Diphtheroides are found on our skin. So just an example, species matters. Some species have, the ones at the top have a lot more tools, have a lot more virulence factors that make them more likely to cause an infection. And like I mentioned before, infectious dose does matter. I read a case study not that long ago, well, it's been a while ago, of a foodborne outbreak at a restaurant. And they decided that the piece of food that everybody was getting sick from was the garlic bread, which is kind of unusual. But the guy that was the sickest out of everybody that got sick had eaten 27 pieces of garlic bread. So he had a much larger infectious dose and he was sicker because of it. So what infectious dose is, it's the minimum number of organisms needed to establish an infection in the host. And that's really variable. Some organisms like this bacteria that causes Q fever, they think, oh, 1 cell can make somebody sick. That's not a particularly serious infection, actually. Others like cholera, you need to ingest 1 billion cells. So there's just a wide variety. Sometimes we have no idea because we don't have a good way to test that. Like it's not ethical to, get a row of volunteers and then, have them drink certain amounts of cholera, obviously. We have a good animal model though, then we know about what that is. And sometimes we express that in terms of kind of the average. So the number of bacterial cells required to establish an infection in 50% of test animals. or sometimes we express that as a lethal dose as well. So there's different ways to express that. Here's an infection that has a really, really small infectious dose. We think that if a person breathes in as little as maybe just a few viruses, they're likely to get this infection. And you've likely never seen anybody with this, but this is what measles looks like. All right, if you have 100 people in a room and all of them are susceptible to measles, nobody has had their vaccine, and somebody walks in with measles, probably 99 of those people are going to get sick. So it's an enormously easily transmissible, or it only takes, again, a few viruses, airborne, to make you ill. Okay, and then factors, host factors that affect virulence. These are the same kind of things that make a opportunistic, or your normal microbiota then kind of turn and cause disease. So, age, young babies, older adults are going to be most of the time more susceptible to things. Babies, their immune system's not developed properly yet. In older people, it wanes. Defects in immunity, physical, mental stress. I would add to that lack of sleep. And I know that hits home with a lot of you, but you don't get enough sleep, then your immune system's not, working quite at full capacity, probably. And there are differences in our immunity as well. So, two people, same age, they're both healthy, get the same infectious dose of the same organism, one gets sick and one doesn't. We have differences in how our immune system presents antigen to our B cells and T cells. So there are differences in that, even if one person is not immune suppressed. Obviously, if you've got some other factors going on, like you're on immunosuppressive drugs from an organ transplant, or you're undergoing chemotherapy or something like that. So all those things will affect your immune health. Okay, so we're going to follow the path of infection. So say, how is individual, how does an individual get infected with some pathogen? Well, usually that pathogen is going to come to a portal of entry in order to be successful in establishing an infection. So these are the places in our body that we're kind of susceptible, I guess, to organisms. And make a note, these are exogenous pathogens. So we're talking about a pathogen in the environment somewhere or on another person. How does it gain access to our body? Skin is not a terribly good portal of entry. Usually organisms are not successful at breaching our skin. There's only a few of them that can actually breach skin when it doesn't have any break in it. I didn't post this, but if you look at a cross section of the skin, we're going to look at this a little bit more closely when we start talking about innate immunity. It's a pretty effective barrier because we have lots of layers of cells, and the cells on the surface are dead. So they're continually falling off. And so pathogens, they attach to it, then they kind of lose hold. Maybe we're a little bit susceptible, with the hair follicle there, but the skin is a pretty good barrier. So it's not a common portal of entry as long as the skin is intact. Most commonly is the mucous membranes. So this is any opening we have to the outside of our body. Again, we're going to look at defenses that we have there in our mucous membranes, but respiratory tract, this is probably the #1 portal of entry. The most common portal of entry for pathogens is the respiratory tract. And probably the second one right behind that is the gastrointestinal tract. So again, we'll explore some of the defenses that we have there, but there are ones that are a little bit more easily breached by pathogens. But then, of course, the urinary tract, reproductive tract, the eyes, those are all considered mucous membranes. Placenta is not as common. We have a good barrier there between the growing fetus and the mother, where usually pathogens can't pass that barrier. There are a few exceptions. The virus that causes German measles, the protozoa that causes toxoplasmosis, lives in cat feces. So that's why if you're pregnant, you should never scoop your cat litter. There's only a few exceptions there. So that's not a common portal of entry. The last one here, the parenteral route. That word kind of looks like parental, but parenteral. So if we break that down, par means against or not, and that root enteric means intestinal. I forget if it's Greek or Latin, but we'll come up with that root a few other times. So basically this means not intestinal, or kind of thought about not the normal route. Okay, so anything that's considered parenteral means that you have a break in the skin. Maybe a person's been stabbed with a knife, or you have an insect bite, or any kind of wound. So if the pathogen enters by something that doesn't normally happen, a syringe maybe, then that's considered a parenteral route, right? Bit by a mosquito, get malaria, parenteral. All right, so those are the portals of entry. Once the pathogen is exposed to the correct portal of entry, then its task is to attach to our cells. And I'll mention that pathogens have to come to a certain portal of entry to be successful. So for example, if we inhale HIV, we're not going to get AIDS. Okay, if we swallow HIV, we're not going to get AIDS. That's not the right portal of entry. It has to be, bloodborne or sexually transmitted. So it has to come to the correct portal of entry. And then if it finds the right cells that it has, the proteins or the structure for, then it will be able to attach. Okay, and that's really the first step in order to establish itself. It's got to attach to the right cells. Organisms are going to have some kind of adhesion factor to help them do that. So we would consider that a virulence factor that helps them do that. We could have a particular structure that enables them to attach. Something like fimbriae, we've talked about that before, or maybe pili. Okay, so those are special structures that bacteria have. So just to review, here's a picture there of a bacteria. The fimbriae are the little short hair-like things that are really important for a lot of organisms to attach to our cells. And there's going to be proteins on the surface of those fimbriae called adhesins that then are going to help them be specific for certain kinds of host tissue. Okay, so in addition to structural things, at least talking about bacteria, bacterial attachment proteins. Okay, we either are going to call those ligands, or another name for that is adhesins. Okay, so makes sense, adhesins, but that term ligand, again, that describes some kind of protein that enables the organism to attach to some host receptor. Okay, so here's a picture in your book here that talks about adhesion factors. I left this for us to label ourselves. So here's the bacteria cell, and here's a host cell. Okay, here's the host cell membrane. This is some kind of host receptor. And then what the bacteria have to recognize the host receptor, we call those, most often they're called ligands. They're drawn a lot bigger here. They're normally not, they're just proteins, so they're not going to be huge like that, but they draw them big just so you can kind of see them and understand there's usually a lot of specificity there. SARS-CoV-2, the virus that causes COVID-19. It has glycoprotein spikes that are going to be specific for certain, now we think they're enzymes that are expressed on our cells. So you might have heard that in the news. They're called ACE2, angiotensin converting enzyme 2. So these ACE2 receptors on our alveolar cells, so deep in our lungs, we have these receptors, and that's what COVID-19 is specific for. So there's going to be some recognition there to help the bacteria or virus or whatever. Here's another example. Here's some E. coli, this kind of grayish part around here. That's a bacterial capsule. So we know that the capsules can help bacteria adhere to structures, surfaces as well. E. coli also have fimbriae, and remember there's adhesins or ligands on those fimbriae as well. So lots of structures to help it attach. Here's another example of a pathogen that it's really important for it to attach to a specific kind of cell. These green cells here, those are Bordetella pertussis, the organism, the bacteria that causes whooping cough. And it's specific for our ciliated epithelial cells in our respiratory tract. And this is an organism that doesn't actually usually invade our cells, but it does have to stick to a certain kind of cells. So we know there's like 8 different adhesins that organism makes, 8 different ways that it stays stuck to its target cell. Okay, so that adhesion is usually enormously important. All right, let's think about some of the tools that an organism might have, or virulence factors, in order to continue to invade deeper into the host once it attaches. Okay, so we're going to look at each of these separately, but 3 virulence factors, extracellular enzymes. All right, another way to say that is exoenzymes. So if you're in lab, you just had a lab a couple weeks ago and tested for 5 different exoenzymes. So these are enzymes that are secreted outside of the cell. The enzymes that we're talking about are not ones that are used just for nutritional purposes, but again, ones that are going to make it more successful in invading deeper into the host. Toxin production. There's a couple of different toxin that organisms can make that make it successful. So we're going to talk about those. And anti-phagocytic factors. Okay, so we're going to just review briefly some steps of phagocytosis and then talk about how some organisms have found a way to actually survive that assault. Almost there. Okay, so you can kind of think of it as kind of a war between the pathogen and the host. So the pathogen has these weapons, and then our host immune system, we have these weapons, and then the pathogen does this, and then it's just kind of that arms race that goes on. All right, so here's a few examples of extracellular enzymes. Okay, hyaluronidase, difficult to say, and collagenase. Hyaluronic acid, we can think about that as kind of, I'll write that up here. Hyaluronic acid is kind of the glue that holds cells together. Okay, so our epithelial cells are the cells that are kind of lining the inside of the body with the outside of the body. Those are held together by hyaluronic acid. So some bacteria can make these extracellular enzymes, hyaluronidase, which basically allows the bacteria to fit between those epithelial cells. So important for invasion. And then collagen, that's a really important structural protein that you find throughout the body. Okay, so if the organism's making collagenase, and then it's able to eat through that structural protein. Okay, so it's all about invasion. It's our chief structural protein. Some more examples. Hopefully one of these will look familiar if you were in lab this week. Coagulase. Right, so if you're in lab, we just tested for that. That's an extracellular enzyme that enables the organism to kind of clot the blood around it. Okay, so if it can do that, it can kind of wall itself off. So you can kind of see that here. It's kind of walling itself off from the rest of the body. So however that bacteria gained access, you know, maybe it was in the bloodstream, maybe it was in a wound, it's kind of partitioning itself off. And that's important because we have all sorts of immune cells that are going to chemotax right to that spot where they sense the whatever bacteria are. And if the bacteria can kind of protect itself for a little while, then it allows the bacteria to grow in numbers. And eventually, when it's you know, using up all its nutrients and grown in numbers enough, it will break out of that clot with an enzyme like kinase, and then be, you know, kind of stronger and more likely to invade deeper into the tissues. All right. So those are some examples of extracellular enzymes. Toxins. Okay, toxins, what do toxins do? Well, in general, they damage host tissues, they harm tissues. There's two different kinds of toxins that I want to talk about. There's endotoxin, which we have mentioned before, and exotoxin. Okay. Endotoxin, if you remember, is part of the outer membrane of gram-negative bacteria. It's actually the same thing as lipid A. So let's go back. Let me show you a picture of a gram-negative cell wall, just to review that. Again, this is only in gram-negatives. Here's the cell membrane and the layer of peptidoglycan. And then here's the outer membrane, and those little blue things that are stuck in there. Here's the lipid A portion of the LPS, the lipopolysaccharide. That's composed of lipid A and some core polysaccharides that are kind of sticking out there. Okay, so that's right in there in the outer membrane. Lipid A, again, that's the same thing as endotoxin. I don't think I posted that, but just to kind of remind you. Okay, so, you know, why do we call it endotoxin when it's actually on the outside of the cell? Well, they originally thought that it came from inside the cell. So it's not a good name, but it's kind of what we stick with. Okay, and again, this is kind of be gram-negative only. Gram-positives don't have an outer membrane. All right. When is this a problem? When does the cell release it? Only when the cell wall is disrupted. Okay, so that can be when the cell divides, maybe to a very small extent, because maybe only a little bit of that cell wall and cell membrane is broken up when the one cell becomes two. Even more so, even more when the cell dies. Okay, so again, even more. Because whenever a cell dies, the whole outside layer of it, the cell wall, starts to break up. And then that's going to release the lipid A. If it's, again, it's causing an infection, it's going to release the lipid A into the host. So whether it dies just by itself, whether we give some antibiotic and it dies, or whether it's dying you know, from macrophages phagocytizing it and releasing kind of the bits of it, then it's going to release that lipid A. What kind of effects does that cause? Well, it's considered low toxicity, but if you get enough of it, it can be a huge problem for the host. It's going to cause things like fever, inflammation. And we're going to talk much more about what inflammation is probably next week or the week after, so stay tuned. But basically, during a huge inflammatory response, your blood vessels get leaky, cells and fluid are moving out into the tissues. If your blood vessels get leaky enough, then you're not getting enough oxygen to your organs, and that can cause shock. Okay, so if you get enough inflammation, a patient can go into shock, and that can be deadly. All right, so again, just a little bit of endotoxin, not a problem, make the patient kind of uncomfortable, but you get enough of it and it's a huge problem. Okay, things like blood coagulation, hemorrhaging, things can be caused as well. Okay, exotoxin, on the other hand, both gram positives and gram negatives can produce exotoxins. And these are secreted proteins. So hopefully you're thinking, well, what's the difference between an exotoxin and an extracellular enzyme? Extracellular enzymes are proteins, so are exotoxins. The difference here is that extracellular enzymes are only going to work locally around the cell that secreted it, so right near the cell that secreted it. Where where exotoxins are going to act systemically. Okay, so they can get in the bloodstream and work all around the host's body. A little bit of it goes a long way. So extremely high toxicity and kills host cells at a low concentration. Botulism toxin, that's an example of an exotoxin. We use Botox, which is botulism toxin, an enormous number of medical applications because we dilute that botulism toxin millions of times. Okay, so a little bit of it goes a really long way. We use Botox as muscle relaxants and treat migraines and all sorts of things now. Okay, but again, it's a little bit of it goes a long way. And typically exotoxins are very specific. Okay, so they're acting on something very specific and have kind of a very specific job. Okay, so here's some examples of exotoxins. These are the ones your book mentions. Something like it's considered a cytotoxin. It's not as specific. That's just going to kill host cells. Okay, so they talk about that. Cytotoxin kills host cells. Not very specific there. Neurotoxins. These are going to interfere with nerve cell function, and of course, nerves determine whether we can move our muscles or not. So an example of this botulism toxin that will act on our nerves so that our muscles can't contract. It's called a flaccid paralysis. So eventually, if you've got botulism, eventually you lose the ability to move your diaphragm and breathe. Tetanus toxin, on the other hand, is also a neurotoxin. It acts a little different way. It's so that our muscles contract and then we can't relax them. Okay, so the patient, that's called lockjaw. Tetanus is called lockjaw, and patient loses the ability to relax their muscles. Enterotoxins, it's another exotoxin. So sounds like endotoxin, but not the same thing. Remember, this prefix entero means intestinal. Enterotoxin are going to affect the cells lining the GI tract. Okay, so Staph aureus, for example, if you get Staph aureus food poisoning, Staph aureus can produce an enterotoxin that gives you diarrhea and makes you throw up. Okay, so here this diagram just kind of compares exotoxins and endotoxins a little bit. Saw some of the effects of endotoxin, and then in this chart in your book, they also compare exotoxins and endotoxins. So you want to kind of keep these straight in your mind. Some of this we didn't really talk about, like we didn't talk about heat stability. Don't worry about that. Fever producing, whatever endotoxins do, typically exotoxins not. We didn't really talk about this antigenicity. That just means, do we have an immune response to it? For exotoxins, yes. So that means we can have good treatment for it. We have vaccines that work against diseases that the primary harm is from toxins, things like tetanus. Unfortunately, we cannot make a vaccine for endotoxin. So that just means we don't have as good treatment for that. We don't have any, antitoxin that we can administer to help with that. Just gives you some examples of diseases that are primarily the harm is from a toxin. Okay, don't memorize these. This one, meningococcal meningitis, the effects from that are mostly due to a really severe variation of endotoxin. And we're going to talk about that one later on in the semester, but again, those are just examples, okay, to help you learn the difference between exotoxins and endotoxins. All right, the third type of virulence factor are anti-phagocytic factors. All right, so what do I mean by that? We'll talk in more detail about phagocytosis next week. Phagocytes, these are our white blood cells that engulf and remove invading pathogens. Okay, so you probably have some familiarity with that already, but we've talked about phagocytosis before. But just to kind of review, a phagocyte, like a macrophage, for example, is going to extend pseudopods around a bacteria and engulf it in a membrane endosome, which we call a phagosome. And then our lysosomes will fuse with the phagosome. Lots of digestive enzymes in lysosomes that most of the time are going to be successful in killing that pathogen. And then, whatever is left over, the bits of it are going to be exocytosed from the white blood cell. Okay, so that's how phagocytosis normally works. But some bacteria have ways to avoid this process. Okay, so sometimes they have virulence factors that are going to help them prevent being engulfed in the first place. One very common one, we've mentioned how many bacteria are able to produce capsules because it helps them survive phagocytosis. Not forever, but it helps them avoid it for a little bit longer. So the host can kind of have some more tools at its disposal. Okay, so capsule production helps them avoid phagocytosis. Another strategy is that some bacteria can produce leukocidins. Okay, leuko meaning white blood cells, cidin meaning to kill. So this is a chemical, a cytotoxin actually, that kills white blood cells by poking holes in its membrane. Another example of an antiphagocytic factor, strep pyogenes can produce this protein on its surface called M protein. And here's a picture of some strep pyogenes that can make M protein, and here's a cell that can't. So again, the M protein, these are on the outside of the cell. That M protein actually keeps some proteins that we make in our immune system called complement, which we're going to talk about soon. Complement is something that attaches to the outside of the cell and makes it easier for our phagocytes to engulf bacteria. Because strep pyogenes have this M protein on the outside of our cell, our complement can't bind to it. So it's one strategy, just a strategy to help the cell keep from being phagocytized. Sometimes cells have, get engulfed, but then have mechanisms to keep it from being killed during that process of phagocytosis. So for example, we would call this preventing digestion. Mycobacterium tuberculosis, that's the organism that causes TB. It's really good at this. It gets engulfed by phagocytes, but then it secretes chemicals that keep that lysosome from fusing with the phagosome. So those mycobacterium, they just kind of hang out and reproduce right inside that cell that's supposed to be killing them. It's a really devious strategy. It makes it hard for our immune system to find them. Okay. All right, so we're just flying through disease here. Stop and think for a minute. Okay, how do we describe how this process is acting on a patient? Okay, so the stages of infectious disease. When we're first exposed to a pathogen, and say that pathogen enters the correct portal of entry and it starts to reproduce itself. So when we first become infected, and we're talking about stages of illness. To begin with, we have, when the pathogen is first being established, it's called the incubation period. Okay, so there's some pathogen in the body, but the patient is not showing any signs or symptoms. And if we use those terms correctly, a sign is something that's observable. So that's something like a rash or fever or vomiting. Where a symptom is something that you feel, but nobody else can tell. So something like headache or malaise, tiredness. Okay, so when we talk signs and symptoms, if we use those words correctly, signs something that somebody else can notice, and a symptom is something that the patient feels. Okay, so in the incubation period, there's organism multiplying, but the patient doesn't know it yet because they're not feeling anything. How long is that incubation period? It's totally variable. It depends on what organism you're talking about. Talking about rabies virus, that incubation period can be up to like 9 months, because it's a time it takes the virus to migrate up to the to the brain, I think. Other things like flu, influenza, incubation period is about a day. We know COVID is somewhere between 2 and 14 days, but normally 4 to 5 days incubation period. Once the patient starts feeling some mild symptoms, signs and symptoms, then we call that the prodromal period. And that might be a term that's a little bit less familiar. So prodromal period is you wake up in the morning and your throat's kind of scratchy, or you just feel just not quite right. Okay, so that's why we're self-assessing every day. So you notice those prodromal symptoms, and then you take that to, oh, do I need to stay home today with COVID? All right, is the pathogen infectious during the incubation period or the prodromal period? Again, it totally depends. Pathogens that are infectious in the incubation period, they're the ones that are really difficult to control transmission. Chickenpox is especially infective during the incubation period. So that's before people feel any signs or symptoms. They're just going about their regular day and they're passing it on to everybody they meet. Of course, COVID can be infectious when patients are not symptomatic. So that's a real problem in controlling it. But a lot of infections are not infectious until the patient starts feeling bad. As those signs and symptoms get worse, that progresses to the illness phase. Most severe signs and symptoms With some infection, the patient dies. It never gets beyond that. But if the immune system kind of starts to win out, then we get a decline period, declining signs and symptoms, and you get a little better every day. And then convalescence is when you're returning to health. You may still be maybe a little bit weakened in some ways from the infection. Okay, so probably the only term there that's maybe something that you would need to review there is that prodromal period. Okay. That term. All right, so portals of exits. So patient is infected. To be a successful pathogen, that pathogen wants to move on to another host. And usually the principle here is generally they're going to be shed from one host in larger numbers than what they came in. So, you know, you might have breathed in 10 viruses or whatever, but you're going to be coughing back out many, many more, okay, to give that pathogen a better chance of infecting another host. So our portals of exit are going to reflect the same ones of portals of entry. So it's fairly intuitive here. Respiratory and salivary portals. So breathing out, even when we breathe, just breathe out, we do often expel some very, very tiny droplets. Those droplets can be carrying virus or bacteria. Okay, so even breathing can be a portal of exit, but even more so coughing, sneezing, talking, laughing. That's why we all have a mask on right now, unfortunately. Fecal exit. So any organism that's considered fecal oral, so you ingest it and then it's passed out through the feces, they are expelled in enormous number, usually in diarrhea. So when you see those words, increases motility of bowel, scary words. Here's an example. I don't know if you've ever seen these. These are cholera cots. So in places, maybe, you know, refugee camps or whatever, where they have cholera go through, patients, they just put patients on these cholera cots because they have such horrendous diarrhea that they can't get up and get to the bathroom. They end up, you know, just soiling themselves over and over again. So they put them on these cots so that they'll just drain into a bucket, basically. So it sounds awful, and I'm sure it is. Patients with cholera often die of dehydration because, again, the fluids are just running through them. So that's the key to treating cholera: hydrate, 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 usually with some kind of IV. Okay, so that fecal exit, that's quite successful for a lot of pathogens, especially in places where they don't treat their water. So, you know, you've got sewage contamination in the same water that they're using for washing and drinking. Your genital tract, sexually transmitted diseases there, bleeding, you know, it's mostly fluids that are going to pass these pathogens. Okay, so once those pathogens leave a host through a portal of exit, where are they maintained in the environment? They might go straight to another host, but oftentimes they can survive in the environment somewhere for a time until they can infect another host. So we call those sites reservoirs. All right, sites where pathogens are maintained. And a reservoir can be an animal, it can be a human. We're gonna distinguish between animals and humans for once, or it can be non-living. Okay, so typically a reservoir, if a reservoir is an animal, it's typically maintained with no harm to the animal. And that's not always the case. For instance, we would consider a dog a reservoir for rabies. Okay, rabies kills dogs. So that's not always in the strictest sense. But something like bats. Bats are a really common animal reservoir. They carry many different diseases. Why do they carry so many diseases? That's something that we don't quite understand, but they live communally. So they live basically on top of each other, in caves and places for the day. And then in the nighttime, they fly, away to faraway places, and then they come back together again. So they're in contact with lots of other different, animals, and they come back together again. Interesting enough, bats live 20 years, which is astonishing. Mice, which are about the same size, only live like a year or two. So I don't really understand why bats live so long, but there is something to our intrinsic fear of bats. So Halloween this weekend, right? They carry lots of nasty stuff. But when we are in contact with an animal that's harboring some kind of pathogen, we call that pathogen a zoonosis or a zoonotic infection. All right, and usually those zoonotic infections are a lot harder on us. So when they kind of spill over from animal populations and infect us, they're a lot tougher on us. We get them when we're in some kind of contact with animals. So either through an animal bite or, hunting the animal or being in contact with their feces. Okay, I see that I'm about out of time. So we will finish up with just a few things here in chapter 14 on Tuesday. Be working on your homework this weekend, so you don't have to do that right before the exam. Hope you have a good weekend.